Good afternoon. Thank you for joining us. I'm Krista Kaups from UCSF Fresno. I am really pleased and honored to be the introducer and moderator for this session. We're talking about resuscitation in subpopulations, new and emerging research. How can we take better care of our patients? As you saw from the announcements, we've got a great panel of people who are participating. And as we've all said to each other, we all look forward to learning from each other. First up is Dr. Jason Sperry, MD, MPH, who is a professor with a primary appointment in the Department of Surgery, Division of General and Trauma Surgery, and secondary appointments in the Department of Critical Care Medicine and Clinical and Translational Science Institute at the University of Pittsburgh. I think many of you are familiar with all the things that Dr. Sperry has been involved in. He obtained his MD from Case Western Reserve, did a general surgery residency at Johns Hopkins Hospital, MPH from the University of Texas Houston School of Public Health, and a fellowship at the University of Texas Southwestern Medical Center. If we started listing his research interests and expertise, it would take up all the time. So I think we'll directly have Dr. Sperry educate us. Thank you so much.
Thank you very much. I think everybody can hear me. I'm going to share my screen and am I sharing? There we go. I am sharing. Okay. Everyone can see first slide. So my topic is going to be resuscitation in subpopulations, new and emerging research.
Jason, you might want to go to the slideshow view.
It's probably we discussed that.
I'm sorry.
It might not work. That's totally fine. I'm gonna proceed. And the focus of the new and emerging research will be on traumatic brain injury. I have no conflicts. And again, a quick overview. I have about 30 slides, so I'm going to try to do this in 15 minutes or 14 minutes. So a quick overview. We're going to talk a little bit about early and pre-hospital trauma resuscitation. And you might ask what the heck that has to do with traumatic brain injury. But then we'll talk about early resuscitation for traumatic brain injury. Sometimes the early bird gets the worm. Also talk a little bit about trials and future directions. And this is like my intro to many of my talks. You know, polytrauma, which is traumatic brain injury and hemorrhagic shock, man, these things don't mix. They're additive, and they create a severely injured patient. The brain injury makes the bleeding worse. The bleeding makes the brain injury worse. And it's just a very difficult patient population, but that's what we deal with consistently. Again, in review, this was, one of the best trials, in the last 20 years. By Dr. Holcomb and 1:1:1, the proper trial, which really brought to the life the 1:1:1 resuscitation, which was sort of demonstrated in the military setting to start with the first trial. Though it didn't hit primary outcome from a statistical significance, it showed separation, and that 1:1:1 was better, and it reduced death from exsanguination. But despite us, this changing practice, the mortality from in this trial and subsequent trials still was extremely high early within the first 1, 2, and 3 hours after traumatic injury. And what this does is highlights the pre-hospital environment. This was a amazing JAMA article by Dr. Stacy Shackelford from the Department of Defense. This is non-randomized, but an amazing study that showed the benefit of pre-hospital blood transfusion within 10 to 15 minutes of severe injury. And these patients had a significantly higher survival, lower mortality, and after propensity score adjustment. One of the early papers demonstrating early, before you arrive at definitive care, even in the Department of Defense situation, in a military situation, that early blood, early is better. And again, highlights the pre-hospital setting for interventions. Really, I want to talk about two trials that were done in a civilian setting. One is the PAMPER trial, looking at pre-hospital plasma and patients at risk of hemorrhagic shock. And obviously, there's a TBI correlate that I'll get into. And then the Resuscitation Outcomes Consortium, the ROC-TXA trial, which was focused on patients at risk of traumatic brain injury. And so I'll highlight these two studies and a little bit of secondary research in the PAMPER trial. So this is probably one of the most important secondary analysis of the PAMPER trial. And we looked at outcomes in patients with traumatic brain injury. About 25%, about a quarter, 20 to 25% of patients had traumatic brain injury in the PAMPER trial. And when we looked at it, we showed that those with no traumatic brain injury on CAT scan, so definitive brain injury, there was no harm signal of giving plasma up to 2 units pre-hospital. There's certainly no harm signal. In the overall trial, there was a 10% mortality benefit. But when you look at those with traumatic brain injury, that mortality benefit increased to around 25%. And that was the driving force. It was in these patients with polytrauma, hemorrhagic shock, and brain injury that a survival benefit was determined by pre-hospital plasma. If you looked in this secondary analysis, if you looked at subgroups, patients with TBI had all the signal, and if you looked at those that had the lowest GCS, less than 8, that were statistically significant, shifting to the left, the sort of the red squares, and it was those patients with severe polytrauma, the ones with the high injury severity score, in addition to head AIS, other abbreviated injury scores that were elevated greater than 3. And so it's the sickest of the sickest with the highest probability of probably mortality that maybe plasma has its benefits, but it also, in a sense, the survival benefit is strongest in TB and patients with TBI. So early pre-hospital may be better. So there's pre-hospital, and then there's early pre-hospital. Well, this was from that same paper where we looked at if you were transported from the scene, the helicopter picked you up in a field, and from the scene or a rendezvous with an ambulance, and you got plasma earlier, closer to the time of your traumatic injury. If you got transferred from an outside emergency department or an outside hospital, you got transferred from an ambulance to an outside emergency department, and then you got picked up by a helicopter and then got enrolled. So the time from injury is presumably longer. And so the signal, again, was those that were transported from the scene. There's also probably a survival bias, that if you're transferred from an outside hospital, that you live to be transferred to a trauma center, and so maybe plasma isn't going to make a difference if you're not at major risk of dying. Though there is a significant mortality in this cohort, plasma was not beneficial if you were transferred from an outside hospital. The ROC-TXA trial, this is extremely important. They had 3 groups. One got 2 grams pre-hospital, another got 1 gram, and then one got nothing. The 1 gram and the 0 are the red and the black lines in secondary analysis in those with documented traumatic brain injury. The cohort was at risk for TBI. They had to have a low GCS, but you can have shocks. So some of those patients didn't have TBI diagnosed on their head CT. So the overall study didn't hit statistical significance, but when they looked at subgroups with documented TBI, there's a strong mortality benefit of 2 grams of tranexamic acid. And my favorite sort of slide is TXA. I put that shit on everything. That is just because it's safe. And there was a whole bunch of worries about it, but it's been shown in multiple trials, and even more recently out of Australia in the hemorrhagic shock cohort, again, that it's associated with the survival benefit. It's small at times, but it is there, and there's no harm signal. So again, if you have TBI, I want 2 grams of TXA as early as possible, particularly in the pre-hospital situation. And the Department of Defense, this has become standard care for in the military setting. You're getting 2 grams TXA. It may not have taken off across every pre-hospital environment, but there are some that are using 2 grams. A lot of them are using at least 1 gram, but 2 grams the Department of Defense has taken a hold of. So early plasma, I'm going to jump back because our group has done some secondary analysis looking at pre-hospital plasma. And I'm going to highlight the first pre-hospital plasma. It's associated with this distinct biomarker expression following injury. And we showed that the patients with the severest injuries had the survival benefit. In a sense, cluster B on this right hashtag. These are the sickest patients, had the worst biomarker expressions, and they had the greatest benefit from pre-hospital plasma. Similarly, we've done global patterns and looked at lipidomics, metabolomics, and proteomics based upon the findings we had in the Pamper cohort. We looked at lipid species, metabolites, and proteins. And really what this shows is very similar to that prior work that was I demonstrated in the pre-hospital plasma and JCI Insight. It showed that there's a sicker cohort, and we call this endotype 2, using multiomics, multiple platforms. And the sickest cohort, they have a higher mortality, they have higher injury severity, they're more commonly injured via blunt mechanism of injury, and they're more commonly have traumatic brain injury. But most interesting is when you look at endotype 1, which is the less severely injured, and you look at TBI or no TBI, there's no difference in either. In other words, in the gene expression and the protein expression and the metabolomic expression is really no difference, and there's no differences across plasma. And then in the endotype 2, the severely injured, more commonly blunt, more commonly having TBI, that it's in this endotype 2, the severest injured, where plasma has extremely strong survival association. Now, these patients aren't randomized, but these are adjusted in models. And we looked at the gene differences, and there are differences in the endotype 2 with TBI versus everyone else. They're a special cohort. And we've sort of done a deep dive. We did multi-platform, multi-omics, and again, looking at this, and we did causal mediation analysis, trying to figure out sort of the independent drivers of what makes plasma beneficial in this endotype 2, the severely injured TBI cohort. And in this, there's certain factors in this mediation analysis. Again, these are the E2 patients was described. Restoration of depleted clotting factors, inhibition of and blocking of protein C and fibrinolysis, promotion of molecules involved with lipid transport, lipid metabolism, mitochondrial energy, and protection of the blood-brain barrier. Things that sort of make sense, that would be positive, but at least they're highlighted by these multiomics. And again, these potential biological implications or the causal mediators, again, it's enhanced clotting. In patients with TBI and severe shock, they bleed, they have an additive effect. And so when we're suppressing fibrinolysis and the consequences of inactive protein C and plasminogen, lipid transport and production of lipid mediators, and brain protection. And that's what some of this high sort of multiomics, sort of expensive biomarkers in different sort of aspects has demonstrated, and that you could be able to predict those patients that might benefit from early plasma. You could do sort of a directed resuscitation in the pre-hospital setting with the right tools and some point of care testing that could predict an E2 patient type and TBI. So just to highlight freeze-dried plasma, we don't have it available in the US, but this is a game changer. You put this on every stop the bleed, have a little thing of dried plasma and some saline, and it has a long shelf life. It can just hang out. You can put it in public schools, and you can give this. You don't need to refrigerate anything. And so this is a game changer, and a game changer for the military, such they're already using it. They're just using other countries' approved products. Where are we with the US? We have pending freeze-dried plasma products cooking as we speak, but they are not approved currently. What about whole blood resuscitation? Whole blood has plasma. Plasma seems to be doing better for traumatic brain injury, and it's safe for resuscitation for non-traumatic brain injury. Well, we're looking at whole blood resuscitation, troop in hospital, lights, tower study, pre-hospital, and again, a whole bunch of observational studies that have showed whole blood's beneficial. And I don't need to, that's not what this talk is specifically on. I did want to comment on platelets and traumatic brain injury. Currently across the country, except since July of 2023, all platelets were stored in room temperature for up to 5 days on a rocker, hung out in your blood bank. They had lower hemostatic capabilities. and they had a longer life. They hang out in the system in your post transfusion. They hang out longer, but it's been known for a long time, for 30 years, that cold refrigerated platelets have a lower risk of infection. They're more robust from a hemostatic standpoint. They have a shorter half-life in the body. The spleen takes them up because they're activated. But if you're bleeding, these might be what you want to assist with bleeding. And I know Julia will be probably commenting on platelets also, but cold store platelets may be beneficial. So why do I bring that up? Lights Network did two trials in cold store platelets. This is well before they were officially approved for blood banks to make it as of July of 2023. We did a one in hemorrhagic shock, which has been completed and published in Annals of Surgery. I won't comment on. And then one in TBI. The TBI was a phase 2. It was 100 patients randomized at the University of Pittsburgh. We completed enrollment in late 2023, and we just finished getting our 6-month Glasgow outcome scores. And really what we were comparing in this cohort, patients that required platelet transfusion with traumatic brain injury, particularly those patients on aspirin. These were an elderly cohort primarily. Cold store platelets were given earlier because they were available in an emergency department fridge, and compared to room temperature platelets, and also they were cold relative to room temperature. So there was two differences, sort of early and cold. The manuscripts in submission process, I am not able to comment, but let's just say there's some benefits associated with cold stored platelets in this cohort. And that'll be for a future sort of publication coming towards you. Can we predict pre-hospital TBI? So you never know whether you're in a pre-hospital setting on an ambulance. How do you know? We've done some analyses looking at a whole blood cohort. We looked at early GFAP and UCH L1. These are point of care TBI biomarkers. And we showed that on arrival, a cut point was very predictive with the ROC curve of 0.76 of predicting TBI. This is point of care light. This is an early ED blood sample. And we also looked at GFAP, a different cut point, and it predicted a worse head CT, and it predicted a worse 6-month Glasgow outcome score and long-term follow-up from TBI. So we might be able to predict. And interestingly, the point of care testing for early TBI prediction was just approved for a whole blood point of care testing. So on a helicopter, on an ambulance, we could send these biomarkers, measure a cut point, and we could predict the presence of TBI extremely early and predict the prognostic at up to 6 months out. This has been accepted in an impress at Journal of Neurosurgery, though it's not in PubMed yet. Again, in conclusion, the earlier the better, the pre-hospital, whether it's plasma, whether it's TXA, whether it's cold stored platelets, whether it's whole blood, and early the better is better for trauma patients in general, and TBI is one of the ones where it's most important. Again, summary, the early bird gets the worm, TBI also, and I would recommend probably for resuscitation for TBI early TXA, early plasma, and soon coming to your knowledge, cold stored platelets. Happy to answer any questions.
Fantastic. That is exciting. I mean, first to start with TXA, which all of us have and use, but just to really encourage us to push that out to our pre-hospital folks if they're not already. We decided in advance, and I'm sorry I should mention it to the audience, if people have questions, I would say go ahead and put them in the chat, and we'll ferry those along. While you're thinking of and you're typing in your questions, the question I would have, just kind of looking to the future, in a few years, are we going to have the ability to figure out the endotype 2 folks? We'll get a quick scan, like a glucometer, in that, and they'll say, oh, it's the endotype 2 folks. That doesn't sound like it's too far out.
I would agree. My chairman, who was a major driver in that data, although he's Dr. Billier, and my sort of partner in crime with those publications, again, based upon plasma data and plasma early Pamper samples. And there's a couple biomarkers, probably 2 or 3, and have a high predictive capabilities of the E2. and with an UCHL1 and some of those TBI biomarkers, the combination of those, and that's going to be changing like concussions in the NFL, concussions, but at the same time, the less likely severe brain injury, but also you could use those TBI biomarkers for the severe TBI and predict the diagnosis of. So the combination of that could be on a little bit of one single point of care testing, and yes, you could do it. Now, I would still say that I'd give whole blood if you got a refrigerator and you could have it somewhere. It's hard to do it on an ambulance. I'd give freeze-dried plasma on an ambulance for sure. So once it comes available and there's no storage requirement. And so there's no harm in plasma, even if you didn't have TBI, you know what I mean? But in a perfect world, most of the signal is in TBI patients. That's where the survival benefits, at least demonstrated in the Pamper trial. So,
and, and I think those, all of us who are involved in taking care of trauma patients, we know what the devastating effects, short-term, long-term are, and just the long-term disability for TBI. And we haven't had a whole lot to offer our patients. So this is really exciting. And it actually seems like it's something that's within reach. So terrific, very exciting. I'm not seeing anything in chat, or Q&A. So if anybody has questions, please put those in there, because clearly we've got an expert. If you don't think of something now, put it in the chat and we'll get to it later. So terrific. All right. Our next speaker is Dr. Tanya Anand, who is a clinical assistant professor of surgery with the University of Arizona. She's also MD NPH, which I thought was intriguing. Of all of our speakers are all MPH folks. Dr. Anand started at USC for her MPH, received her MD at St. George's, did a residency at Kern Medical, and a fellowship at the University of Arizona. And she's going to talk about the impact of aging on post-traumatic endotheliopathy, etc. And since we're all aging, I think we are inherently interested. And as our patient population seems to be getting older and older, we've got our 90-year-olds, I know, hanging out in the ED right now on our service. So without taking any more time, Dr. Anand.
Thank you for the introduction, Dr. Kaups. Yes, it's a mouthful, this title, but essentially, I really wanted to give a brief roundup of what's been going on with aging research, endotheliopathy, as well as the metabolome. I have no disclosure that pertain to this particular talk. So when we look at the overall care we're giving to patients, we're caring more and more for injured older adults, and it's become a significant part of our trauma care delivery now. In the US, in a 10-year period, there was an increase from 18 to 50% in the number of patients we're really treating that are 65 and above. I mean, if you look at this little picture from the Census Bureau, essentially by 2035, there's a prediction that adults 65 and above will outnumber individuals 18 and below. And that's significant. And this number is only expected to rise and further influence the trajectory and the emphasis that we have on our trauma care in this country. When we look at the most common mechanism of injury, the most common mechanism seems to be low mechanism, which is a ground level fall. But the impact it has on these older adults is not low. I mean, these patients suffer from very significant injury from these low mechanism falls. And there are a number of studies that are showing that increased age and frailty are associated with worse injuries with similar mechanisms. With aging, there's an increased morbidity and mortality and an increased case fatality rate for individuals with similar ISS scores, but different age groups. In addition, end organ complications significantly spiked in individuals 45 and above. And that doesn't seem like a very, high age, 45, but that's a common inflection point seen in some studies that looked at age and outcomes. When we look at this, the special nuances and the needs of this group, this led the ACS to, recently update the National Triage and Inpatient Management Guidelines. Given that we have special considerations in triaging and inpatient management because of their unique physiology for the older adult, should resuscitation considerations differ in this group as well? And when we look at resuscitation over some 200 years, our practice has slowly become more and more focused as we understand the physiology of resuscitation better, blood banking, and the impact that each product has on the patient. Resuscitation goals now are not just improving perfusion and replacing volume. There's other considerations now, such as minimizing the coagulopathy and the endotheliopathy that results after moderate to severe trauma. And this is reflected in the evolving landscape of publications in trauma and resuscitation. If you look at the last 10 years, especially in PubMed literature, you better understand that there's likely more to transfusion than simply volume replacement. And it's how we manage these other things, the coagulopathy, the endotheliopathy, that helps the patient's trajectory after resuscitation. And so we're going to talk a little bit about the other stuff, the endotheliopathy and the metabolome, especially in older adults, and how it may differ to their response in younger adults after resuscitation. So when we talk about the endotheliopathy of trauma, the first question comes to mind is, what is it? It's really a global description of the systemic endothelial injury caused by trauma and or hemorrhage that leads to disturbances in certain areas, such as coagulation, the actual barrier between the vasculature and the tissue, vasoregulation, inflammation. And it all comes down to really specifically the inner layer that lines the vasculature is that glycocalyx and the breakdown of it and how it exposes the underlying endothelium to further insult. And it starts immediately after trauma. This is a picture of an intact glycocalyx layer. Those fine hairs protect the underlying endothelium. They kind of regulate that space there. It's an anti-adhesive. It repels the white cells and the red cells and attenuates platelet binding. It also helps regulate permeability, signal transduction, vascular reactivity, and overall and modulates what the underlying endothelium is exposed to. Normally, young and middle-aged adults have this full head of hair, so to speak, of these proteoglycan complexes. But then during trauma, what tends to happen is this barren wasteland occurs, depending on the severity of the trauma. And when this is disrupted, it causes not only a local and a regional response, but it causes a systemic response as well. And this response ultimately leads to increased risk for the patient to have a coagulopathy, a higher transfusion requirements, and greater risk for multi-organ failure. And greater injury severity has been correlated to this greater disruption. When we look at the body of literature that's out there that has explored the impact of the endotheliopathy of trauma, we see that it does have a significant impact. That greater inflammatory and cytokine response and the presumed disruption of the glycoproteins lead to further dysfunction and multi-organ failure. But when we think back and look back at it now, older adults tend to have worse outcomes than younger patients. And is this the case in terms of the endotheliopathy of trauma? Do they have the similar reactions, similar types of shedding patterns? Is it the same between older and younger adults? And we were curious and we sought to explore this by taking another look at the PROPER trial. The PROPER trial was a prospective study that enrolled severely injured patients who required massive transfusion. And we performed a secondary analysis of this trial. And the aim of our secondary analysis was to really characterize endothelial glycocalyx injury in severely injured older adults and compare it to younger adults, and to determine associations between advanced age and the putative regulators of glycocalyx shedding. We included patients who were bluntly injured, primarily because we felt that the vascular endothelium in those patients would be more likely to be subject to more injury and more susceptible and vulnerable. We stratified patients in terms of age based on quartiles. So a young adult was the lower quartiles, the older adult population was the upper quartile for age. And we did unmatched and matched analyses of these groups. And in the matched analyses, we wanted to really control for the influence of injury and shock severity on Syndican 1 shedding. And we'll talk a little bit about what that is. We ended up with 350 bluntly injured patients. Of those patients, 258 were young adults and 92 were older adults. And this is a very, very busy slide, but it's primarily showing our matched patients that we compare. The last column on the right is the older adult, the middle column is a younger adult. And the older adults were significantly older in the two groups. And they also had greater degree of comorbidities. The injury severity scores and the shock index was pretty much the same between the two groups. When we looked at vitals upon presentation, older adults tended to present, and it was significantly different between the two groups. They presented with a lower systolic blood pressure and a lower heart rate, higher hemoglobin levels, and lower platelet levels. And these were all significant. There was no difference in the amount of crystalloid and amount of blood products that were administered to these individuals. Now, when we look at Syndican is a surrogate marker for glycocalyx shedding. We measure those levels, and it gives us an idea of whether or not potentially the glycocalyx is shedding more, is disrupted more. And that's the primary proxy we use for that. And we see here that at presentation in hour 6, older adults had significantly less Syndican-1 plasma levels than younger adults. And in terms of the regulators of shedding, the putative regulators such as high mobility group box and TIMP2 or tissue inhibitor metalloproteinase, in older adults, they were significantly lower than younger adults. And then we also noted that the levels between the shed ACEs or the shedding regulators correlated with plasma syndican levels at hospital arrival. In terms of outcomes in the matched population, the older adults had significantly less vent-free days, significantly less ICU-free days and hospital-free days. They trended towards a higher mortality both at 24 hours and 72 hours, but was not statistically significant. In terms of our regression analysis, we found that the increased age was independently associated with decreased levels of the certain shed ACEs or shedding regulators, even after controlling for gender and presence of comorbidities. When we looked at the relationship between age and syndican levels, that relationship was even a little bit more dramatic in the sense that as a person ages, the syndican levels on presentation were significantly lower or were lower as they aged by 3 times. And so there were several takeaways from this particular study. One, that age seemed to be independently associated with decreased circulating syndican levels, when in fact in the literature up until this point, age wasn't really a factor we've seen. We've seen higher syndican levels associated with higher amount of injury, but in this case, age was associated with decreased levels. Putative regulators of glycocalyx shedding were also associated with age, and there was a correlation between age and the shed ACEs as well as the shed ACEs and syndican levels. And what we can say from this is that the suppressed plasma levels of these regulators in the setting of diminished syndican suggests that there's a mechanism regulating the cleavage that may be impaired in older adults compared to younger adult patients. And when we look at older adult patients, I mean, this is just one study of many that has shown that there's some differences between older and younger adults on the molecular level. And there are several studies out there that have shown a difference in terms of both noradrenaline, adrenaline levels, anticoagulation, hyperfibrinolysis. And in the second study, there is a significant signal of immunosuppression in older adults versus younger adults in severe sepsis and septic shock, even up to 28 days post the septic event. In terms of coagulation in TBI patients, the same stands for D-dimer and fibrinogen levels. That not only the initial presentation, but the change in several hours is significantly different in older adults compared to younger adults. And this evidence doesn't just extend to endotheliopathy research and coagulation research. More evidence is noted when we examine the metabolomics literature. Metabolomics is a systems biology science that really is focused on identifying and quantifying small metabolites or small molecules such as sugars, nucleotides, amino acids, and lipids. And really it is focused on trying to better understand the phenotypic signature of that individual that stems from the genome and transcriptome and proteomic response to the environment. And more info is gotten as more research is performed on this. Well, we have found in older adults in the aging metabolome that trauma has a certain impact on those patients, but these patients coming in the door, older patients coming in the door compared to younger adults, tend to have a pre-existing oxidative stress inherent, which potentially can make recovery from the similar trauma different between an older and a younger adult. The next several articles really talk about the fatty acid redox and glucose pathways in older adult alters with age or as a person ages, such that a metabolic age score would be potentially more accurate than the actual numeric score of age itself for an individual. And finally, we're starting to see that the relationship between the metabolic response to trauma, they could be a driving force to the endotheliopathy and coagulopathy we're seeing after trauma. And with this slowly accumulating evidence showing that aging is playing a role in how an individual responds to trauma, perhaps the aging metabolome may be impacting the different reactions we're seeing in glycocalyx shedding, as well as the shed ACE reactions to trauma that occurs in older adults. This is a study that I think that Dr. Sperry just cited as well. We have to question, why is this all important in terms of resuscitation? If you look at patients given a similar treatment, certain individuals, certain endotypes, or certain subgroups of individuals tend to respond differently based on their endotype. And in his study, which he had just cited, there's endotype 2 patients with TBI and multisystem trauma tended to benefit from early plasma compared to endotype 1. And so this is important, and this may be the case with different older adults as well, with different injury patterns. There's already evidence that early utilization of plasma and adjuncts, such as tranexamic acid, may have an impact on the patient's recovery and the amount of resuscitation they end up getting after a trauma. But these studies are primarily performed in middle-aged and young adults. What about older adults? How does trauma appear to be affecting their metabolome and the glycocalyx, and how does this data apply to them? Does it depict the disruption in the same way? And we've already seen to some degree that there's some evidence that they may not be the case, that the disruption impacts them differently. And I think with more time and more research, it will kind of facilitate our ability to answer those questions a lot better. So a few takeaways from this: the injured older adult has a unique physiology and a pre-trauma state when they walk in the door compared to the younger adult. And at least that's what the evidence seems to start saying. Whether this aging metabolome is impacting the patient's post-traumatic response and recovery, that remains to be ascertained. And finally, when we talk about balanced resuscitation, what is balanced? Is it different for an older adult? Is it different for an aging metabolome? And I think that requires a little bit of investigation as well. And that's all I have. And thank you for this opportunity.
Wow, that was exciting, and glad to hear that. And again, I don't think we have any questions or chat. Caitlin, are you helping keep track as well? So please ask questions, and we'll be glad to take those. The thing that always occurs to me with these is, and we know that as we see patients, that there are patients, the throwaway line is age is just a number. And, but there are people who physiologically are older at 40 than other people are at 60 or 70. And I wonder if we'll have a way to really assign out old physiology versus young physiology. It makes me think about that. Dr. Anand, any comments?
I mean, if you look at how we see frailty, that's like a phenotypic depiction of kind of what's already had started on the molecular level far before. So if a person appears to be frail at 75, when did that process start on the molecular level? And I think that's where metabolomics and studying that a little bit better may help us. If you see some of the studies that are out there, when we look at older versus younger individuals and their outcomes, there's this interesting inflection point seeing at the age of 45, I'm seeing in multiple studies. And it's much younger than we thought. We're like 45, ooh, that's young, but maybe it's not at a certain point for a certain individual. So it's interesting, and it remains to be studied more, I think. We need more people looking at this.
It's part of that. So thank you very much for broadening our horizons there. And our third speaker is Dr. Julia Coleman, MD, MPH, who is at The Ohio State University. We're kind of coming full circle. As we mentioned, Dr. Sperry started in Ohio as well, but Dr. Coleman also did graduate and medical school studies, at Ohio State and the University of Toledo, then went west to Colorado for residency, came back to the Ohio State University for her fellowship, and now is finished fellowship and is out and about, and has a ongoing interest in many different things, I know, but we're going to hear about sex dimorphisms and coagulation. So the sex-specific considerations in transfusion. So we don't have to just think about whether they've got a brain injury, how old they are, but we have to think about sex as well. So more things to think about and maybe better ways to treat our patients. So thank you so much, Dr. Coleman.
Thank you so much, Dr. Kaups, and the AAST for the opportunity to speak today. This is really, I think, an exciting topic, and really a privilege to be a part of the panel of other speakers as well. So I'm going to talk to you all after I reveal that I unfortunately don't have any disclosures. about sex dimorphisms. So sex dimorphisms exist in nature. In birds, one can most readily distinguish between males and females phenotypically. These same drastic dimorphisms exist in coagulation, with females demonstrating a relative hypercoagulability as compared to males. They form clots faster, at a greater rate of clot propagation, increased clot strength, and decreased clot breakdown. And why does this matter? Because it makes a difference clinically. Our group first looked at all severely injured trauma patients at 2 level 1 trauma centers and found that female-specific hypercoagulability persists after injury. And in the setting of trauma-induced coagulopathy, female sex confers a survival benefit. So, I'm not the first one to find this. There's been a lot of people over the years that have looked at the role of biological sex in impacting outcomes in trauma and have found a differential outcome with female trauma patients. So I'm not the first one to write about this. And in fact, in the proper study, which has now been discussed by all 3 speakers, just recently, findings from that study were published that revealed that females receive lower blood product transfusions after anatomic hemostasis. So this continues to be demonstrated across studies. And so the question is, what is the mechanistic explanation for this bedside observation? And how can we take those understandings of the mechanisms back to the bedside to impact our resuscitation and transfusion medicine? So that's what I'm going to be talking about in 14 minutes tonight. So again, when you look at the overlapping TEG tracings, which is a tool that we use to look at hemostatic capacity in whole blood, you can see that there are a myriad of differences in the coagulation cascade by sex. For one, we know that there's shorter clotting time in females. This is an effect of different thrombin biology. Female sex is associated with higher levels of circulating factors, shorter conventional coagulation times, and this comes down to even the level of proteomic analysis. We've already talked about omics tonight as a powerful tool in research. And if you look at the proteomics between males and females in their plasma, there's a remarkably higher level of coagulation factor 5, which is corroborated with activity assays. We also know there's higher soluble tissue factor in females, including in pathologic states, and a myriad of other proteins that we know converge on thrombin biology are also different by sex. There's greater amounts of complement in females, decreased antithrombin. What's driving this? What's the mechanism? Part of it we think is driven by sex hormones. And the reason for that is because we see shorter clotting times in women's on in vitro fertilization with elevated therapeutic hormone levels, increased coagulation factors in women on OCPs, in pregnancy, and during peak estrus of the menstrual cycle. So estrus cycling and estrus levels seem to correlate with that increased thrombin generation. So shorter clotting time, that's one, but another is increased clot propagation, which you know is mainly an effect of fibrinogen and fibrin. And we know this is also different by sex. Female sex is associated with higher circulating fibrinogen and higher functional fibrinogen that we look on a functional fibrinogen TEG. And when you even look under a microscope, dynamic fibrin formation and elastic modulus is higher in females. So the clot structure itself is different. And not only is that clot structure by fibrinogen and fibrin biology different in formation, but also in its breakdown as well. Female sex is associated with increased fibrinolytic resistance, including TPA resistance and increased levels of Pi1, both of which we know are powerful players in the fibrinolysis cycle. And so, just like with thrombin biology, we think this is also partially driven by sex hormones. That's because female sex is associated with increased fibrinolytic resistance, as mentioned. But if you look even in murine models, so overrectomized rats have higher levels of fibrinolytic activity. Premenopausal females, so higher estrus levels, have higher circulating Pi1 as compared to postmenopausal females. And in the setting of OCPs or oral contraceptive pills, there are robust increases in Pi1 and decreases in fibrinolysis. So we think this pattern, again, is also driven by sex hormones. So there's a difference in the fibrinogen, fibrin, the fibrinolysis. The last thing we haven't talked about are platelets. And this is really my area of interest and research. We know that clot strength is mainly an effect of platelets. And platelets are super interesting because not only are they highly decorated in many, many types of receptors, they also express estrogen receptors, which I didn't know until I started doing research into this area. And so we know that, first of all, there are differences intrinsically in the platelets of males versus females. If you take ADP and you stimulate male and female platelets, the female platelets go wild. The male platelets don't do that much. the opposite happens if you stimulate platelets with platelet activating factor. And not only are there intrinsic cellular differences in how platelets respond to agonists by sex, but we can also manipulate that with sex hormones. So if you take male and female platelet and you stimulate them with ADP, the female platelet goes wild. The male platelet doesn't do that much. But if you treat that male platelet with estrogen, then that feminized male platelet's behavior looks the exact same as a female platelet. So certainly there's intrinsic cellular differences, but sex hormones are playing a role as well. And how does this happen? It's interesting because estradiol is a sex hormone receptor. And we know hormone receptors are nuclear receptors. Translocate down to the nucleus, they affect transcription. But platelets are anucleate cells. So how is it that a hormone receptor could be changing the biology of a platelet? And that's because there's quite a bit of research that has characterized non-genomic effects of estrogen. So estrogen can actually impact translation and change signaling pathways inside cells that is totally apathetic to the nucleus. And so we see in vivo a same phenomenon of estrogen playing a role in a platelet hyperactivity. We see increased platelet reactivity and elevated estrus in vivo. And there's also research that describes differential response to Plavix by sex. So we think that there is a sex hormone effect. So I keep talking about estrogen. Let's talk a little more about it. So estrogen, we know in vitro and in vivo, causes a hypercoagulability. If you take whole blood and you spike it with estrogen, it shortens time to clot formation, it increases clot strength, decreases fibrinolysis, increases functional fibrinogen, and as I mentioned, increases platelet reactivity. And we see the same thing in vivo. So we've done research where we followed women prospectively throughout their menstrual cycle, and we have correlated peak estrus with these same hypercoagulable changes. Moreover, we've conducted prospective studies in women through pregnancy and see these same changes with increasing estrus during a pregnancy. But how? How does estrogen affect all of these changes? There's a variety of ways. One is that this non-genomic effect of estrogen I talked about means that there are hundreds of proteins that are affected in their translation and their signaling, their phosphorylation, all of that within cells just by estrogen. If you add estrogen to whole blood in vitro, it causes increases in hundreds of proteins after just 15 minutes of incubation. And a lot of those have to do with coagulation and platelet biology. And if you add estrogen to whole blood and form clots in vitro, and then look at the cross-links between the fibrin monomers, estrogen in females causes a significant increase in multiple cross-links between the fibrin monomers. Each of those depicted here, the Fib-G in green, Fib-A in red, the Fib-B and blue. Mainly, we see increased cross-linking between the Fib-A monomer. So it is changing proteins, it's changing cross-links, and surgeons have to see things to believe them. You can see this when you look at the clots under the microscope. If you add estrogen to whole blood and to plasma and you form clots in vitro, the estrogen causes an increase in clot resolvability index, which is essentially clot density. So you're forming a more highly structured, distinct, dense clot. So that's a lot of basic science and benchside work. How do we take this back to the bedside? I could spend a whole talk about really what I think are the mechanisms of these non-genomic actions of estrogen and platelets. A lot of it has to do with intracellular signaling cascades like SERT kinase, but I don't want to bore you all tonight. So let's get back to why does this matter for patients? How do we take the fact that we know estrogen is affecting all of these hypercoagulable and pro-platelet changes? How do we take this back to the bedside? Because if you think about this data, you have to wonder, should we be thinking about this with transfusion? When you hang platelets, do they come from a male or female? Does that matter? Well, our research suggests that it does. So we said, okay, let's see if anyone's looked at this yet. Well, in fact, people have already started looking at concordance and discordance in vivo in transfusion and found that there's a difference in outcomes based on the concordance of the donor and recipient transfusion. The study that was recently published looked at polytrauma patients receiving transfusion and found that FFP and cryo recipients were more likely to experience adverse outcomes when there was a sex discordance between the recipient and the donor. So the sex matters. We said, okay, let's look at this in vitro. So we did transfusion in vitro, where we took blood in a coagulopathic model, and then we took blood from males and females, and then we added blood products from males versus females in sex concordant and discordant pairs, and found that platelets have a more hypercoagulable effect and female fibrinogen has a more hypercoagulable effect. So the female blood products are evoking a greater restoration to hemostatic capacity. And if you look at the cryo of females, it has a higher association between total and active fibrinogen when you look at the functional fibrinogen and correspond that to the fibrinogen levels on mass spec. So the blood products are different, and it matters clinically when you're transfusing them. This was recently presented at AAST and was published by Margot, one of my mentees. Shout out if she's on this call, she's amazing, and is a budding star in the world of trauma. So female and male coagulation, it's not the same, and it's partially mediated by intrinsic differences in the machinery of coagulation, but also by estrogen itself. And so again, this calls into question whether we should be thinking about donor sex. When you hang platelets, I want you to start asking yourself, did these come from males or females? And does it matter? Because I think that it does. And maybe we should be monopolizing on that and strategically transfusing by recipient sex or donor sex, because that matters. But also, potentially, there's a role for increasing the efficacy of the platelets that we transfuse by treating those platelets with estrogen pre-transfusion. So spiking them and creating a super platelet. Now, you might think this is crazy. You're saying we're going to give sex hormones to trauma patients when we're resuscitating them? This has been looked at before, not just in treating blood products, as I mentioned, but actually giving patients sex hormones. This was looked at before and actually in trials, randomized control trials, looking at progesterone and spinal cord injury because of progesterone's ability to mitigate the secondary insult of neurotrauma and inflammation. So this has been looked at before. And we know in the world of trauma, when you have a traumatic injury, there's an increase in your endogenous estrogen levels. And we know that estrogen has an ability to mitigate the insults of shock, of inflammation, of organ dysfunction, and particularly of coagulopathy. Now, someone that's been a pioneer in this work has been Irshad Chowdhury, who is a professor emeritus at UAB and has done some incredible work in animal models looking at, in trauma, what would it look like if we were to start giving estrogen as a therapeutic adjunct when we resuscitate patients. He published a murine model of traumatic injury and hemorrhagic shock where male rats were given estrogen, and it significantly improved cardiac performance and attenuated trauma-induced inflammatory marker release. He also did work in which he did a large animal model of TBI, coming back full circle to the beginning of the webinar here, where estrogen was given IV, and it increased the cardiovascular performance, it decreased acidosis, and it improved survival. In another swine model, this one of polytrauma and hemorrhagic shock, animals were randomized to escalating doses of estrogen, and it increased 6-hour survival and survival time. So there have been other areas where they have looked at other pathologies where they've looked at giving estrogen therapeutically. They've looked at this in transplant, in on-pump bypass, in uremic bleeding, in GI bleeds. So using estrogen as a therapeutic adjunct has promise not just for trauma patients, but any bleeding patient in the hospital potentially. So again, this all comes back to how can estrogen help to mitigate the coagulopathic response to trauma, and whether we can potentially, in my dream of dreams, treat platelets with estrogen to create a super platelet that will have an increased efficacy when we transfuse it in our trauma patients to improve survival. So I want to acknowledge my mentors in my coagulation core. Rich Gamina is an interventional cardiologist here at Ohio State. Tom Hund, who is a biochemist at Ohio State, the director of DHLRI, which is where my lab is housed. That's where I am right now. I got some platelets that I'm working on as we speak. Bryce Curlin, he's a pediatric hematologist at Nationwide. Mitch Cohen, a trauma surgeon at University of Colorado, and Mackie Neal, a trauma surgeon at Pittsburgh, both of who have been great mentors to me. Christy Townsend, a neurobiologist at Ohio State that studies sex hormones, and Xiang Lang Liu, my amazing tech in my lab. This is my team. It was also on my KO8 grant that I submitted this morning, so shout out to them. And I would love to entertain some questions. If any of you are interested in this topic, reach out to me. You can read this review we just published in the American Journal of Hematology about the implications of all of this for the care of our trauma patients. And here's my contact information. Would love to entertain any questions.
Oops. Julie, Dr. Coleman, that was fantastic. And I just, as I'm thinking about all this, and I'm thinking about our trauma patients, I can see their trauma cocktail is expanding. We're going to be studying them better, and we'll give them what we call the PET cocktail. It'll be we'll give them plasma, estrogen, and TXA as we get going with that. No, it really, in many ways, we start out as we take care of trauma patients, as you look back, say 10, 20 years ago, so many of the things we do are fairly crude, and we hopefully make progress as we've progressed in the last 20 years, a better understanding of what we're doing as we resuscitate patients and take care of them. Sorry, I thought I had my video on here. And that is exciting. Is there a difference between pre and postmenopausal patients? I mean, that's the question that has been asked many times.
Yeah, that's a great question. So we've done some work of this looking at pre and postmenopausal female platelets. And one would imagine that if this was all just related to estrogen and circulating sex hormones, that presumably postmenopausal females would have platelets that were not as hyperactive because they have decreased circulating sex hormones. But that's actually not the case. Some of the work that we're doing shows that they are more hyperactive. And I think the reason for that points out this is not just about sex hormones. There are intrinsic cellular differences. And not only do platelets express estrogen receptors, but megakaryocytes express progesterone receptors, estradiol receptors, and androgen receptors. And my hypothesis, which will probably be something that we look at in RR, is that does a lifetime of your bone marrow being bathed in sex hormones and change the platelet lineage? And I think that's very possible. So it's a great question, and we're still looking at it.
Excellent. Again, our participants must all be really very well attuned or very well or very knowledgeable about these subjects. I've got to think there's got to be some questions. And I also will open it up. Panelists, do you have questions for each other? So Dr. Anand, Dr. Sperry, if you're able to, we've got a couple of minutes, so let's open up the floor, because I think we, again, do best when we learn from each other.
Yeah, I actually, if Dr. Sperry is still on, I noted in one of his slides, he was talking about metabolomics markers as indicating the presence of a TBI or not, without even knowing, without having the scan. Are there markers that are present that kind of indicate progression of a traumatic brain injury, like worsening, that can be predictive for worsening pathology or TBI?
Yeah, great question. GFAP, glomerular fibrillary specific protein. So on the study that we did, it was a secondary analysis in a whole blood study that was published in the Journal of American College of Surgeons. So a certain 15% had traumatic brain injury. So we looked at these biomarkers, and the first cut point, which was 286. That was what the study showed, was the best predictor. That predicted a diagnosis of TBI. A cut point of 3,000 predicted worse Rotterdam scores on serial CAT scans, and then worse 6-month Glasgow outcome score extended. So the higher cut point demonstrated worse outcomes relative to the lower cut point, in those patients with TBI. And then we did a multivariate analysis because we included patients without TBI to see what was the prediction. We looked at any confounders, head and neck AIS, extremity AIS, abdominal AIS, shock severity, injury severity score, and it did not affect the predictive capabilities of that biomarker.
That's remarkable, because some of the patients, just anecdotally, we know that there's some patients who have a delayed progression of a bleed, and the only way you catch that is if they've clinically worsened, and sometimes it's too late by that point. And I'm wondering if there's a different marker available where we can utilize that concomitantly with diagnosis, but also predictors of worsening.
Well, that predictive capability is in the first sample in the ED. It's not at 6 hours or 12 hours. That was the first one cut point. You could probably use serial, and that's probably coming our way with this new FDA approval of the point of care testing.
Very
It's going to be studied here probably relatively soon.
cool. Thank you so much.
Other questions? We are at time. Yeah, we see the future. The future is now. So I hope everybody really enjoyed these both exciting, challenging, and also very understandable for the rest of us who are not immersed in the world daily. Thank you for all the participation, and thank you for joining us. The session is recorded, correct, Caitlin? And will be available on the AAST website. So if you've got people who should have been listening, but weren't, or maybe they were in the OR, please direct them that way. So thank you all very much. And we thank you for your time. Wish you a good evening. So,
Thank you.
all right.